Jai Hind all of you. So today we are going to start with the topic features of DNA. So let us start this features of DNA, what it is actually. So in this we are going to see the silent features of the DNA. So let us see the first point. The DNA has antipolarity, which is either 5 prime or 3 prime, or it can be 3 prime or 5 prime. For example, you can see this structure of a DNA. As you know that DNA is a double helical model, so here as you can see that this particular helix, which is over here, is starting from here and getting end over here. So this is the 5 dash or 5 prime, and this one is the 3 prime. Now just opposite, the other helix of this particular DNA is over here 3 prime, and here this is 5 prime. For example, if you want to understand this in a much simpler way, you can study this as if this is one helix and this one is the other helix, then this particular helix can be 5 prime, and then the edge or the last one of it is going to be 3 prime. Now this is the other helix of it. If you will make this DNA strand straight, then this one will be the 3 dash or 3 prime, and this will be the 5 prime, right? Now let us see the next one. We are having the other point as the backbone of polynucleotide chain is made up of alternate sugar, that is your ribose or deoxyribose, phosphate group, right? So which is made because of the nitrogenous base pair plus your sugar group. So because of that, the phosphate group which is added over here, the nitrogenous base projects inward. So nitrogenous base projects inward, which means your purine and pyrimidine, which are over here, right? They are inside the helix of DNA. That is why the word inward has been used over here. So this is one of the silent feature of the DNA. Now let us see the next one. The DNA is in right-handed fashion fold. Means if you will see the structure of DNA, you will see that this is in your right hand side, it is always twisted. Okay, so you will observe that DNA is always folded in the right hand side. So that is why it is right-handed fashion fold it is having. Next we are having feature of DNA is a large size of a purine comes opposite to the smaller pyrimidine. So purine is next to pyrimidine. Now in this, what is there, as we have already gone through and we have studied that there is the pairing of nitrogenous base pairs in the DNA. For example, if this one is over here, one of the BP is going to be here, the other one here, the other one here, the other one here, right? So this is how it is going to get arranged. And we have also gone through that nitrogen base pairs are of two types. One is purine and the other one is pyrimidine. So it is having its some best friends. Purine can be with him its own, right? Pyrimidine can be its by own, but no, it doesn't get through their own structures or types. They make their best friends. Like A, that is your adenine, it has made its best friend T. Guanine is there, right? So guanine has made its best friend C, which is your cytosine. So adenine's best friend is thymine and guanine's best friend is cytosine. So always in this DNA helix, wherever there will be the base pairs pairing, there will be A will be always with the T. Here if A, then here is going to be T. If this one is G, this is going to be the C. This kind of arrangement is done. Now let us see the next one. We are having the fifth one. Pitch of helix is about 3.4 angstrom. Pitch of helix is 3.4, it is 3.4 nanometer and 34 angstrom. Okay, so pitch of the helix, That means when you're looking forward, when you're watching the structure of DNA, so here you will see this folded structure, right? So this folded structure which you are observing over here, this complete folded structure of one twist, one turn of this helix is your one pitch of the helix, which is about 3.4 nm, or you can also call it as a 3 angstrom. This is a really very important question as per your board. This is often asked and a straight question is asked from this one point, right? And this one also makes a very important question. These two are really very important questions of your this particular topic. Now let us see the next point which we are having in the features of the DNA. Here we are having the plane of adjacent BP, planes of adjacent BP, BP is same, your base pair which we always talks about, are staked over one another. How you have seen ladder, आपने सीढ़ी देखी है, आपके घर की सीढ़ी हो या आप बांस की लकड़ी की सीढ़ी देखोगे तो उस पे क्या देखते हो आप, जो स्टेप्स हैं वो वन बाय वन है ना, बिल्कुल एग्जैक्टली एक के ऊपर एक के ऊपर एक ऐसे होती है, राइट? सेम इज ओवर हियर इन द डीएनए ऑल्सो. इसके ठीक ऊपर दूसरा, इसके ठीक ऊपर दूसरा, इसके ठीक ऊपर दूसरा, right? So this is how they have got arranged. So for this purpose we have used the word staked over one another. Okay, so they are always parallel, one by one they are arranged in a DNA helix. Now next point is DNA is negatively charged, right? So now if this is asked that what is charged DNA have, so you can say that DNA is negatively charged and it is having a acidic behavior. So DNA is acidic too. Okay, so DNAs are negatively charged and they have the acidic behavior. Now for the proper functioning of it, it always needs a positive charge, right? And this positive charge is there because of the protein, and the type of a protein is called histone. So there is a histone which is coiled with the DNA with a positive charge and DNA is a negatively charged. And as you know that the opposite charges always attract to each other, right? So because of that it functions in a much better way. So this was the silent features of the DNA. Now let us see some more features. Apart from this, there are some differentiation in all these helix. Alright, so let us see this one. We have gone through that the distance between two BP is the 0.34. So where you can see the distance between two base pairs, this one is about 0.34. Fine. And the number of BP in the turn is 10 BP. For example, as you can, if you want to calculate this one. So this will be having total 10 base pairs. 1, 2, 3, 4, 5. So this is how it is going to be 10 base pairs in one turn of it. Okay. And already we have discussed the pitch is having 3.4 nanometer. Now if you would have to find out or do the calculation that if you will be given the value of A or T, for example, if just A's value is given, so with that you can identify the percentage of guanine or cytosine present in the DNA by using this particular formula. In our next video, we will do the calculations also, that how to calculate the percentage of these nitrogenous base pairs present in the DNA. But for today, this much is there. So hope that features of DNA is clear to you all. Thank you for watching the video.